Hey, everybody. I am here to talk to you about cancer and chemotherapies. So, in order to do that, the first thing that we're going to get into is, well, what is cancer? Cancer cells are cells of a host that have lost regulation of cell growth and proliferate uncontrollably. So essentially what that means is they grow and they copy, they just make more of themselves in an uncontrolled manner. They're also invasive and form what are known as secondary tumors. Grab my pen. There we go. Now, proliferation is the division and growth of a cell. So not only the replication, but kind of the growth, the size of that particular cell. Now, a neoplasm is a new growth or development of a tumor. A tumor is only considered cancerous when the cells in that tumor are invasive. So a growth, if it's kind of limited, is not necessarily a problem. So here what we have is a normal duct, and what we have within that duct is we have the lumen, which is the interior of um, a region of the body. Then we have a benign tumor versus a malignant tumor. A benign tumor, and that word benign you've probably heard elsewhere, that's kind of doesn't do much. Or maybe let me rephrase that. Doesn't, I'll put it in quotes here, cause a problem. Now, in contrast to that is a malignant tumor. Now, what you need to notice about these two different tumors is we have this basal lamina, which is kind of the coating around this. And in our malignant tumor, these cells right here, they don't kind of stay in that one location. They don't stay in the lumen, but instead what they do is they kind of cause an outgrowth. And so these cells, they take on different shapes. And that's one thing that I want you to recognize about this is that if we compare our benign tumor and our malignant tumor, okay, so this is, well, that's uncontrolled cell growth. But those cells are kind of being, or they're remaining within that basal lamina. Whereas in a malignant tumor, we've got those same cells, but then we have all of these other ones that are kind of expanding beyond that basal lamina. They're becoming invasive. They're growing outside of that cell or outside of that region. Now, to think about that, or to think about a parallel there, as I always think about like an invasive species, these in malignant tumors, these cells are kind of an invasive species in some respects because they're going outside of and they're being introduced to a new area, and effectively that's causing a problem. Now, what are the hallmarks of cancer? Cancer cells are able to evade the body's immune system because they arise from host cells. So it's almost like the body's immune system recognizes them as like a part of the host. They're essentially have the same identification, which appears healthy to the body because they're like, oh, well, that's just a liver cell. I've seen liver cells done. That's just a part of the body. Now, the characteristic of cancers includes immortality. What that means is that these cells are going to be able to replicate going on and over again, kind of unchecked. Sustained proliferation. So what that means is they're replicating and growing, and there doesn't appear to be any stoppage of it. And then they evade apoptosis. So apoptosis is a process known as cell death. Now, this is kind of like actually as an example, like if you ever have, dry skin or something like that, that's dead cells or dead skin cells. And so death is kind of a normal part of a cell's life cycle. And, just as like you might get dead skin cells on your scalp or something like that, or maybe, I don't know, just as an example, um, those skin cells are going to be like lost and then replaced with new ones. Well, cancer cells kind of avoid that. And so basically, rather than, my skin cells dying and being replaced by kind of the next generation of skin cells, if they're able to evade, or another way of putting that, avoid apoptosis is these skin cells that are arguably supposed to die don't. And so then, this layer of skin cells that is supposed to replace a set of skin cells, well, the ones that have been brought up to replace, they simply don't because the old ones didn't die. And instead, this process just keeps on kind of accumulating over and over and over again. It's almost like a conveyor belt where the newly produced or the old thing is never taken off that conveyor belt. And so everything just kind of accumulates and, grows and grows and grows. In addition to those characteristics, cellular motility and genetic instability. The cell is more likely to obtain mutations. Now, that's kind of interesting because many cancers arise from mutations, but that's also something, it's almost like the thing that brought them about is going to kind of keep them around. So this mutation that leads to a cancer is going to lead to more mutations. Now, this neoplastic progression, a new cell's progression, well, we've got our hallmarks, the mortality, genetic instability, sustained proliferation, evades apoptosis, altered metabolism. So they're going to process nutrients differently. They can lead to greater inflammation, evade immune killing. They can be angiogenic. What that means is they'll produce new blood vessels and basically give kind of blood new places to go, which, if your body has that could cause cardiac issues. That might cause your blood pressure is going to be thrown off, or your heart rate beat might be distorted because of this. They're going to resist growth suppression. They can lead to an invasion. Now, when those cells ultimately metastasize, we have some hallmarks of metastasis. So this is that kind of that problem place in a cancer. Motility and invasion into different tissues. They can modulate their microenvironment, plasticity, and colonization. So it's almost like these cancer cells, they can, liver cancer, for instance, when it metastasizes, liver cancer can lead to lung cancer. And when that metastasizes, or sorry, when that liver cancer has metastasized, it goes to the lungs, it goes to the brain, it goes to the bones, it goes to the blood. And essentially, we have this uncontrolled growth in many different locations on a person or within somebody's body. Now, to visualize a couple of so to understand what's going on here, I think that the best thing to do is look in this upper left-hand corner where we have what is our primary tumor. A cell goes through a process known as detachment and breaks away from that tumor. Now, what that cell is going to do is going to go through a process known as intravasitation. And that's where it's going to ultimately make its way into the bloodstream and become a circulating tumor cell. Now, what's significant about that is ultimately where that circulating tumor cell is expressing a protein known as CD24, or sorry, CD44. Now, CD44 is a protein that's very significant for cancers because this is involved in the progression of various cancers and can contribute to cancer growth, metastasis, and resistance to therapy. In many cases, higher levels of CD44 expression are associated with a more aggressive form of cancer. Now, that protein is expressed and ultimately binds with another protein known as E-selectin. Now, the significance of that is that this cancer cell that has CD44 expressed will go through a capture, rolling, and firm adhesion process. So basically, we have this protein interaction right here, and then that protein interaction becomes more significant, and then the cancerous cell here associates and it adheres to an endothelial cell. So we've got this endothelial cell, strong adhesion, and then this cancerous cell is ultimately able to go through a process known as extravasatation. So whenever that extravasatation can take place, then this singular cancer cell can ultimately lead to the formation of a secondary tumor. So this would be an example of metastasis, where one tumor ultimately becomes a second tumor. So one tumor leads to the production of a second tumor. Now, when it comes to cancer genetics, there are genes known as oncogenes. So if you've ever heard that term or that prefix ONCO, maybe think of like an oncologist. That's a doctor who specializes in cancer. Oncogenes are cancer-related genes. These are genes that code for proteins that promote characteristics of cancer, like cell proliferation, cell growth, and are mutated to allow for overactivated functionality of those proteins. Now, this is what is known as a gain of function mutation. So essentially, think about this as, you know, everyone has everyone has the gene in question. But for some people, that has a mutation that effectively makes it an oncogene. There are also tumor suppression genes. So whenever it comes to cancer genetics, what I want you to know about are oncogenes and tumor suppression genes. Oncogenes are kind of the pro-cancer genes, whereas tumor suppression genes are genes that code for proteins that block cell proliferation or cause cell death. So these cause apoptosis and are mutated to where they are no longer active. These are loss of function mutations. Now, in normal cell division, what we have is a tumor suppression gene is going to basically regulate the cell cycle. Now, when the cell cycle is under control, well, we have normal cells emerge. Now, if a tumor suppression gene has a mutation in it, then it's going to lead to the abnormal cell cycle, or a cell cycle that is abnormally taking place. Or we could have a mutated oncogene. Now, that mutated oncogene is going to be your gain of function mutation, which is going to lead to the production and accumulation of cancer cells. So essentially, a malignant or cancerous cell division is going to come from either a mutation in a tumor suppressor gene or a mutated proto-oncogene. Now, the most common tumor suppressor mutated in cancer is a protein known as p53. So, a protein called p53 is largely responsible for stopping cancerous cells from proliferating in the body. So, p53 basically it's almost like it's like cancer law enforcement. Oh, that's turning into cancer. Stop right there. Now, cells that exhibit harmful behavior or extensive DNA damage are taken care of by either DNA repair or they undergo cell death. Now, in cancer cells, p53 is mutated to where it can no longer function properly and block cancerous cells from growing and invading other tissues. So mutations in p53 leads to, well, it doesn't function as that regulator of cancer proliferation. So mutated p53 says, okay, cancer is just going to take place. Now, essentially what we have here is in a stress cell, if you look at this kind of model on the right-hand side, almost the middle, basically, we have DNA damage. Now, normally whenever DNA damage occurs, well, that DNA damage can get repaired, and that repair is going to prevent cancer from taking place. Now, p53's involvement in this, well, p53 is going to it's going to undergo something known as phosphorylation. So when p53 is phosphorylated and when it's upregulated, it's going to be able to assist in repair of DNA damage. So what we have here is, if you look back here, so we have our DNA damage. Now that DNA damage is going to lead to the activation of a couple of different proteins. And you just need to know, or you don't need to know these names, but their names are ATM, ATR, and DNA PKCs. So these are all proteins. Now, ATR will activate. It's almost like it's turning on a light switch of this protein known as CHK1, and it turns it on by phosphorylating it. Phosphorylated CHK1 can then go on to phosphorylate P53, effectively activating P53. So essentially, ATR recognizes DNA damage and says, uh-oh, I better turn P53 on. But the way that I have to do that is by turning on CHK1. Now, CHK1 will then phosphorylate P53, making it the active form. This other group, DNA PKCs, what they are able to do is they are able to upregulate P53. So they'll also recognize DNA damage. And when P53 is activated, essentially it can go through all these different processes. It can activate and turn on either DNA repair, apoptosis, cell cycle arrest, senescence, which is again kind of that normal kind of winding down of cellular activities. Ferrotop, ferroptosis, differentiation, autophagy, stemness. It can also, sorry, P53 can block stemness, metabolic reprogramming, and invasion and metastasis. So essentially, P53, whenever it's activated, it can turn on the expression of lots of other genes that lead to tumor suppression. So, in summary, P53 is good. You want a non-mutated P53 because that P53 is able to turn on the expression of lots of different genes in order to suppress tumor growth. Now, DNA tumor viruses. Okay, viruses can also cause tumor progression and cancer development. So tumor progression and cancer development in the body. Viruses that do this are called DNA tumor viruses. A DNA tumor virus accidentally integrates their DNA into the host cell. Now, because viruses are highly active in replicating, they cause the host cell to proliferate as well, thereby causing cancer in the body. Okay, so viruses are highly active and they cause the host cell to proliferate. So they basically say, hey, cell, I need to be activated. I need to be replicating. So you need to replicate. Papillomaviruses are an example. Let me not cross that word out, but papillomaviruses are an example of DNA tumor viruses that can cause cervical cancer in women. Now, this is why medical professionals strongly encourage all peoples to take the HPV vaccine because this can prevent papillomaviruses, which effectively will lead to the formation of cancers. Now, the way that this all takes place is viral proteins mediated for controlled replication of the virus. The host chromosome here and the chromosome of the papillomavirus. So these viral proteins are there so that they can control the replication of the virus. But what they ultimately do is that this viral genome gets integrated into the host chromosome. Now, this unregulated production of viral proteins leads to and drives cellular proliferation. So it's almost like this virus, HPV as an example, could potentially do nothing, but it could also cause cancer. And the way that it causes cancer is by basically the integration of a viral DNA fragment into the host chromosome. And whenever that happens, then all of these viral proteins are like, oh, well, I need to replicate that. And so they jump on to, the familiar part and they say, well, I'm going to replicate this. Oh, and I'm just going to keep replicating. Well, since that's integrated as a part of the genome, it just keeps going and replicates and works to replicate the whole thing, leading to a malignant tumor. Now, there are a number of different ways to treat cancer. Surgery for removal of a tumor, radiation therapy to try to reduce the size of a tumor, chemotherapy, immunotherapy, hormone therapy, targeted therapy, stem cell therapy. These are all kind of on the table. And there are tons of different, very um, influential and positive and successful stories at at the MD Answer Cancer Facility and Cancer Treatment Center in downtown Houston, or the Mayo Clinic in Arizona or in Minnesota. There's tremendously great opportunities to treat cancer with innovative approaches. And many times, people, when presented with a cancer diagnosis, they kind of get it all hands on deck. Well, let's remove this, but then maybe we can't remove the entire tumor, but then so we're going to do surgery, and then we're going to do radiation therapy, and we're going to do chemotherapy, and we're going to do immunotherapy. Personally, I'm thankful that I've not had any sort of cancer diagnoses, but my mother has, and um, she's had a couple of different diagnoses, and she's had a surgery to remove part of uh, a lymph node. Then she had chemotherapy as well as immunotherapy. And, she was treated at the MD Anderson Cancer Clinic. And so they had a very innovative approach. She was a part of a clinical trial. And the clinical trial wasn't like, hey, we're going to try this new drug. But instead, the clinical trial was um, evaluating, well, what's more effective to do the immunotherapy and then the chemotherapy, or to kind of intersperse it, basically, what's a new strategy to treat her types of cancer. And thankfully that has been very positive and it's been successful in her lifetime. So that's really great. Now, chemotherapy. Whenever it comes to chemotherapy, well, it's a chemical. A chemical is going to be used in order to treat a cancer. Now, one of the most commonly prescribed chemotherapies, and think about that word, chemotherapy, a chemical therapeutic. One of the most commonly prescribed chemotherapies used to treat cancer is known as an alkylating agent. Okay. Now, these chemotherapies cause DNA damage to cancer cells, which can cause the cell to no longer be able to replicate and die. Wait a minute. They cause DNA damage to cancer cells? Well, when you cause that cancer or when you cause DNA damage, you know, it's possible that DNA damage was what caused the cancer in the first place. But the idea is that you have this cell that is going to proliferate, it's going to replicate over and over again in completely uncontrolled fashion. You use this alkylating agent and you're going to cause DNA damage. And so then you're going to prevent any sort of replication. Now, one of the drugs or one of the chemotherapeutic agents is known as cisplatin. It's a fun word to say, not a fun thing to be associated with. But cisplatin is an example of an alkylating agent. Now, what cisplatin does, for starters, this arrow right here indicates that it is able to move across a cell's membrane. Now, that's an important thing because think about a cell as basically or a cell membrane as like a barrier. And barriers, we're thinking about a cell that's largely spherical. You can't really jump over it. You can't go under it. You have to go through it. Cisplatin can move across the cell membrane, and whenever it does, well, it gets hydrated, means a water molecule is added to it. Now, if we look at cisplatin, PT is platinum, NH3 is nitrogen and three hydrogens. We got two of those, and then we got chlorine and chlorine. Okay, when cisplatin moves across the membrane and this water is added to it, one of those chlorines is replaced by water. And that gets you cisplatin monoaquat aquated. Then, well, a second O2 or water molecule is added. and it replaces that other chlorine. So now we have two water molecules, in which case what we're ending up with is cisplatin diaquatid. Okay, now that cisplatin or that diaquatid cisplatin is in the cell and it's basically able to find a target. Now, what it is going to do, and if we think back to types of interactions, H2O loves to hydrogen bond. Wait a minute, stop the press right there. DNA, in a DNA molecule, if we look at the center of a DNA molecule, what you will see there are hydrogen bonds. So a DNA molecule is held together by hydrogen bonding. So I'm going to say that H bonding basically stabilizes or holds together. I'm going to say holds. And then I'm going to write DS or double-stranded DNA. Now, what's going to happen is that, well, this water or these waters will hydrogen bond with this G and G. Those are your nitrogenous bases. So nitrogenous bases of DNA. You could just look at this as these H bond or hydrogen bond with, I'm just going to say DNA bases. Now, there are a couple of different ways that that can appear. One is known as an intrastrand adduct. The other is an intrastrand crosslink. Now, what's significant about both of these is this platinum and these NH3s, they're basically protruding out. It's almost like a weird new part of the DNA molecule. Now, what this is going to do is any of the machinery that is there to go and replicate pieces of DNA, it's going to encounter that and kind of hit a wall. It's like I can't copy it anymore because I see this weird thing here. Now, one of the challenges is that normal cells, so non-cancerous cells, will experience cell damage as well if they're exposed to alkylating agents. Okay, so that's cell damage. Okay, so that is kind of a risk, which is one of the reasons why cancer therapy can be so taxing on someone and chemotherapy can be so taxing on someone. If you've ever been around somebody who's experienced or who's undergone chemotherapy or radiation therapy, my grandfather, for instance, had chemotherapy, and I remember the first day that he received that treatment, my family went to visit, and he was just like, he was exhausted. And this was someone who I basically, as long as I had known him, I'd never seen him tired. And so it was kind of alarming. But, this is something that it's almost like it's an acute kind of discomfort, sickness, whatever. But it's very taxing. Now, in addition to chemotherapies, there's also hormonal therapies. Hormonal therapies are medications that stop or slow cancer growth that need hormones to grow. Okay, so let's go ahead and look at this hormone therapy and anti-hormonal treatment. We have CC for cancer cells here. I'm going to change the color that I'm circling because I don't want to write red on red. Cancer cells, cancer cells, cancer cells, and cancer cells. Okay, so we've got CC or our cancer cells. H for hormones, E for estrogen, which is an example of a hormone, and tamoxifen. Okay, so we've got this context, and then let's go back over here. So hormone therapy travels throughout the body to find the target hormones. Okay, so hormone therapy is medication that is basically going to seek out a particular hormone. Now, different types of hormones work in different ways. Makes sense. Different hormones, different kind of responses, different actions are needed. Now, what they can do is they can stop the body from making the hormone so they can block that hormone from being produced. So if there's a hormone that's causing a cancer to develop, well, cut the hormone off at the source, and then you arguably don't have to worry about that hormone causing cancer to grow. Next up is to block the hormone from attaching to a hormone or to cancer cells. Okay, so we can not necessarily block the hormone from being produced, but instead just kind of serve as like a, defense. Stop, stop, can't bind. Alternatively, you can alter the hormone so it doesn't work like it should. Okay, so you can change that molecule. So here what we have, going back to our model, we've got hormones can stimulate cancer cells in some types of oncology. Now, if we're looking at these different models, remember that E was estrogen. Okay, so here what we have is our cancer cell interacting with estrogen. Now, as an example, let's take estrogen as a hormone in the drug tamoxifen. This is used to treat certain type of breast cancers. Okay, so effectively, estrogen is presenting the problem. Estrogen is kind of causing that breast cancer to develop and that breast cancer to continue to grow. Okay, so here what we have is hormone therapy consists in blocking the production of certain hormones or in blocking the action of certain hormones. And essentially, when you have a cancer cell right here that is interacting with estrogen here, 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 and here, well, tamoxifen can replace and kind of interact with the estrogen binding site on that cancer cell. So if estrogen is causing the problem, if estrogen is causing the cancer to continue to develop, and tamoxifen is able to bind to the estrogen binding site, well, what's that going to do? That's going to cause estrogen to not get to where it needs to be to cause the problem. So tamoxifen just kind of blocks it off and says, no, you can't come here. And that prevents that cancer from kind of developing. Now, let's talk a little bit more about hormones. Hormones are the body's messengers. They're responsible for controlling and balancing nearly every major system within the body. Now, hormones impact your growth and development, control your mood, determine how you handle stress, and influence how the body breaks down food. Now, they are produced in the endocrine glands and travel through the bloodstream to various tissues and organs. Each hormone has its own specific goal, carrying a key message to certain parts of the body. For example, the pineal gland produces and releases melatonin. Now, this circulates through the bloodstream and cerebrospinal fluid around the brain where receptors will detect these hormones. Now, higher melatonin levels signal the body that it's time to go to sleep. Now, some of the other hormones that you're probably familiar with. Okay, so here's melatonin. We've got estrogen. This controls sex drive in both men and women and regulates the menstrual cycle in women. So estrogen is not unique to women. It's present for men and women. Progesterone. This influences the body's changes through pregnancy. Testosterone controls sex drive in both men and women. Now, estrogen and testosterone are both hormones that I think more typically people will say, well, testosterone, that's for men, and estrogen for women. But there's a balance. There's sometimes referred to a T/E ratio. Cortisol controls stress. Serotonin controls sleep cycles, appetite, as well as your mood. Growth hormone controls the reproduction of cells and their subsequent growth. Leptin is a hormone that was discovered in the past 30, 35 years or so. Let's see, it's 2024. I think it was in the mid 90s. And this controls appetite and it signals when you're full. Ghrelin is another appetite controlling hormone and it signals when you're hungry. So leptin and ghrelin kind of work to say, hey, eat or no, stop eating. And regulation and control of those is something that is very important in studies and understanding of obesity. Insulin responds to sugar in the bloodstream. And that's something that if somebody is afflicted with diabetes or insulin sensitive diabetes or insulin deficient diabetes, they have to kind of watch their blood sugar and or get injections of insulin. Now, your endocrine glands and hormones. Okay, so there are a number of different endocrine glands: testes, ovaries, pineal gland, pituitary, adrenal, pancreas, thyroid, thymus, parathyroid, hypothalamus. Now, all of these different glands are going to produce different hormones and kind of respond to different hormones. For instance, the pancreas produces insulin, one of those hormones that's involved in sugar metabolism. Well, lo and behold, glucagon is another hormone that's involved in sugar metabolism. Now, the endocrine glands and hormones, well, this is just kind of a greater expansion upon what those are. The gland and the hormone that is present, and or the hormone that's produced, or maybe the primary hormone that's produced, and basically where that is targeted to. So, as an example, the thyroid gland produces thyroxine. Well, the target of thyroxine is the liver. And what that's going to do is control your metabolic rate. Adrenal glands, well, your adrenal glands are going to produce, and I always thought, well, I think my first time I heard this word, I heard adrenal, and for some reason I just kind of intuitively said in. Well, adrenaline. Well, that's going to be produced by the adrenal glands, as is cortisol. They have tons of different sources, and these are your fight or flight and anti-stress hormones. The pancreas produces insulin and glucagon, and these, your target is the liver, and they're going to control blood sugar levels. Now, if there's something that I want you to take away from this, I'm going to put a big star by this. The pancreas. The pancreas produces insulin and glucagon, and it helps control and regulate your blood sugar levels. Now, hormones of the posterior and anterior pituitary gland. What I want you to see here is just kind of the interplay and the complexity of hormones, what they regulate and how they are regulated by different components. So if we take the thyroid gland, the thyroid gland produces thyroid hormones. Okay, so let's just kind of follow that. It's almost like we're doing a connect the dots or like a, what's it called? A maze. Thyroid hormones produced here. These hormones feed back to the anterior lobe of the pituitary gland and the hypothalamus. Well, the hypothalamus is going to then go on. And so it's almost like the thyroid gland is controlled by the thyroid stimulating hormone, but it also influences the activity of the anterior and posterior pituitary. So this, it's almost like a feedback mechanism and just goes forward and backwards simultaneously. Now, diseases that cause hormonal imbalances, there's a number of them. And if we look at the center of all of it, well, your parathyroids on the posterior surface of the thyroid gland. Well, you can have diabetes, hypothyroidism, hyperthyroidism, congenital adrenal hyperplasia, Cushing syndrome, Addison syndrome, Turner syndrome, Prader-Willi syndrome, hypoparathyroidism. All of these things, all of these different disruptions can cause, well, kidney stones, psychosis or paranoia, which I think typically whenever I think of a hormone, I personally think about, well, something that's happening almost like a movement, something tangible, as opposed to a psychosis or a paranoia. I mean, that's deep within the recesses of someone's mind, but it's connected to chemicals. Now, bone decalcification and pathologic fractures can occur. Peptic ulcers, calcium deposits, and pancreatitis. Heart failure can be associated with thyroid disruptions and thyroid issues. Now, treatments for hormonal imbalances, well, one treatment is hormone therapy. Simply provide the hormone to the body or stimulate hormone production in the body through medication. So, as an example, well, if somebody is afflicted with diabetes, they might take exogenous or synthetic insulin. That is a form of a hormone therapy because what that is able to do is that is their body's not producing insulin. So if they supplement, if they inject insulin into their body, then what their body can do is basically all the cells within their body can suck up glucose, suck up sugar from your veins and break it down. Now there are hormone blockers. Hormone blockers are medications to stop production of certain hormones. Methimazole for hyperthyroidism. Now, the endocrine system contains nine major glands and organs that produce, store, and secrete hormones. So, here are those different components. Your endocrine system, if you have any sort of disruption of your endocrine system, you're going to see an endocrinologist, and they're going to test and evaluate all of the different glands within your body. Is there one of these glands that's disrupted? And, the thing is, these are all important glands because of the fact that they kind of play into one another. This is an endocrine system. This is I always kind of think about it like an ecosystem. All of these things work together. They work, and I like this phrase, they work in concert with one another. So if your thyroid gland is disrupted, well, then that can lead to parathyroid issue, which can lead to thymus issue, which can lead to ovaries and testes and hypothalamus and pituitary gland. So making sure that all your hormones are kind of in balance with one another is a very important part of, um, making sure that your body is functioning properly. And there are instances where hormone therapy, whether it's insulin or estrogen, are important for people to have, happy, gainful lives. Okay, so there's a lot to cover here. Now, some of the important points of this, I'm going to jog back and just kind of walk you through again. Um, I want you to know the hallmarks of cancer. I want you to know some of the hallmarks of cancer. I want you to also know the hallmarks of metastasis, specifically thinking about that colonization, basically this process right here, where we have a primary tumor that, well, a cell detaches. And then what that's going to do is ultimately be able to make its way to another part of the body and develop again into another tumor or a secondary tumor. I want you to know what the difference between a benign tumor and a malignant tumor is. Your benign tumor is the one that's kind of contained. Yeah, it's a problem. It's not great, but is it spreading? And this is something that, if someone has a type of cancer that metastasizes, that presents a problem because where there was that uncontrolled growth, now there's uncontrolled growth elsewhere. If you think about like a part of your body, if you have some sort of cancer that's like at the tip of your index finger, is that going to cause a tremendous problem? Probably not. Um, or potentially not. It's got to be monitored. And maybe what you're going to do, or maybe what your doctor will order is, well, let's perform a surgery so that we can remove the tip of your finger. In part because that part of your finger you might not need necessarily, but also, if the cancer is right there, well, we can remove this, and effectively we've removed the cancer through a surgery. But then your physician and your team of oncologists and professionals, they're going to monitor, they're going to, scan. How did we detect the cancer the first time? And okay, well, is there something that we know that caused that cancer to develop? Because we don't want it to go from here to here and then up your arm and to your lungs and your bones, because that's metastasis where it's basically become a huge problem. So I want you to know the difference between a benign tumor and a malignant tumor. I want you to know these hallmarks of metastasis as well as hallmarks of cancer. I want you to know this life cycle, basically, what are the steps from a primary tumor to a secondary tumor? Then I want you to know about P53. P53 regulates a lot of stuff. And as long as P53 is functioning well, then you've got tumor suppression. When P53 gets disrupted, then you're disrupting tumor suppression. Now, DNA tumor viruses, I want you to be familiar with papillomaviruses that are examples of DNA tumor viruses. So these can cause the production or can cause tumors to develop. Now, I want you to also know the 7 different methods, and these are, primary methods for cancer treatment. I want you to know what an alkylating agent is. I want you to know about hormonal therapies, what you can do. And, if I were to take one of these pictures, I'd want you to be able to explain, oh, this is what this is doing. As for hormones, I want you to know, if I prompted you with a definition and gave you different hormone options, I'd want you to be able to select the correct hormone that is associated with the definition. And I think for the most part, these are things that you might hear about on a semi-regular basis. And you might have semi-regular basis, like you've heard of them before. I think most people have heard of these before. Leptin and ghrelin might be a little bit different. You maybe haven't heard of those. So I would definitely brush up on those. As for the endocrine glands and hormones that they produce, I want you to know about the pancreas, what that produces, and what those two hormones do. And then after that's all said and done, I want you to know what insulin does. And um, yeah, that's about it. At any rate, I hope this is helpful. If you have questions, please don't hesitate to reach out to me. I'd love to talk and, you know, break down any points of confusion or anything like that. But I hope you have a great day, and I'll see you later.